Hello, welcome to part 5 of our biotechnology series here. In this one, we're going to look at another really common biotechnology tool called PCR. And I rewrote this lecture because there were a lot of changes in the textbook. And one of the things I wanted to incorporate, since we're in this remote learning environment because of COVID-19, I wanted to show you how COVID-19 is actually the testing that is done for COVID-19. Right now, the gold standard in testing is a PCR test. And PCR is used to detect specific DNA sequences. So it's used quite extensively in diagnostics. Its real purpose is to make lots and lots of copies of a gene. We want enough copies of genes so that we can manipulate it or see it or use it in research or detect its presence. And it is so sensitive and so accurate that it's the perfect type of test for diagnostics. And we use PCR in order to run this particular process. There are a few things that we need. We need first the DNA sample that we're going to test for. So this could be a mixed sample. What's beautiful about PCR is if we get if we know the sequence, the general sequence of the DNA that we are looking for, we could have a mixture of DNA from multiple different sources, and it will literally find that single piece of DNA that is unique to a specific source. So I need, of course, a sample of DNA that I'm going to be looking at. I need RNA primers. Remember, primers are popping up quite a bit in genetics because primers are kind of those little homing beacons. So we need a set of RNA primers. These primers are going to run from 5' to 3' on both strands. So one primer will line from left to right, and the other primer will line up on the other end of the gene from right to left. So we have one primer that'll adhere to our top strand, another primer that on the opposite end of the gene will adhere to the bottom strand. And then those two primers will initiate DNA synthesis using a specific polymerase called Taq polymerase to replicate that DNA in between the two primers. And the DNA that's in between the two primers is that unique sequence or that unique piece of DNA, that piece of DNA that is only present in whatever sample it is that we are looking for. We, of course, are synthesizing new pieces of DNA, so we need to have A, T, C's, and G's available. Those are the dNTPs. All of this is going to occur in a buffer solution so that everything is at the proper pH and such. And we need a machine called a thermal cycler. This process is done by heating up the DNA to make it single-stranded, cooling it down so that the primers can anneal, then maybe warming it up or cooling it down to make sure that the DNA polymerase can synthesize the new strand. Then we will cool it down again so that strands will re-anneal together, and so we have double-stranded DNA again. The thermal cycler will go through these processes. Now, what's really great about PCR is we could diagnose, right now, the PCR they are using in the hospitals can diagnose COVID. They're looking specifically for DNA sequences that are found from COVID. COVID is an RNA virus. So they reverse transcribe the RNA into DNA and then use the PCR to then look for those DNA sequences. And in that particular case, they can diagnose COVID in a matter of hours. Okay. So I'm going to show you a quick video that shows you how polymerase chain reaction works.
The polymerase chain reaction is a method for making many copies of a specific segment of DNA, starting with a very small amount. This technique can be used to identify specific microorganisms from a small amount of DNA and to identify persons involved in crimes from DNA on cigarettes or in a single hair follicle. The DNA to be amplified is mixed with deoxyribonucleotides, a thermal stable DNA polymerase called Taq polymerase, and DNA primers. The DNA primers hybridize to the ends of the gene to be amplified and provide a starting point for the Taq polymerase. The mixture is heated to break the hydrogen bonds in the DNA, forming single-stranded molecules. The mixture is then cooled sufficiently to allow the DNA primers to anneal to each end of the segment to be copied. Taq polymerase then synthesizes the complementary strand of DNA using the primer as the starting point. The temperature is raised again to separate the DNA strands and then lowered sufficiently to allow the primers to attach. Taq polymerase now synthesizes another set of new complementary strands. This process is repeated until enough DNA has been produced to be identified or used for further research. After 21 cycles, one molecule of DNA can be amplified to over 1 million copies. This amount of amplification can be achieved by running the reaction overnight in a thermal cycler, an instrument that automatically raises and lowers the temperature at appropriate time intervals.
Now, at the time that this video was made, they're saying overnight. Now, with thermal cyclers, we can do this in a matter of about 2 to 3 hours. So a test can actually be run in 2 to 3 hours. That can tell us if a particular DNA sequence is present. And if that DNA sequence is present, we know that the organism that sequence is specific to was present in our original sample. Right?
The polymer.
So this PCR testing for COVID, just the other day when I was rewriting this lecture, I went and found this is a price list for a set of primers that are specific to COVID-19. So those primers that they were showing you in that video, you can see that there are these 5 primes. So we have different sets of primers. F stands for forward, R stands for reverse. So they already know that there are specific DNA sequences that are reverse transcribed from COVID, and we can purchase a set of primers for, look at this, about 15 bucks. So for $15, we can buy a set of RNA primers, and that provides us with a DNA length of anywhere from 20 to 24 base pairs. This process purification is through what's called HPLC. It's just how the primers are synthesized and purified. And here OD stands for optical density. This is a measurement that determines how many, gives us a general idea of how many copies these primers will yield using the thermal cycler protocol that is provided with them. So you can see there are lots of different primers that are available. Now RNase here is used for the reverse transcription. So the PCR testing, there's a couple of different kinds. We have PCR, which stands for polymerase chain reaction. We have what's called RT-PCR, which is what they're using for COVID testing. And RT-PCR is reverse transcriptase PCR. And in reverse transcriptase PCR, they first start by introducing RNases that will reverse transcribe a reverse transcriptase enzyme that will reverse transcribe RNA into DNA. And this is done with COVID because the COVID-19 coronavirus, this particular virus, is an RNA virus. The virus carries RNA, not DNA. So we have to reverse transcribe that RNA first into DNA so we can then use the PCR testing to look for the DNA sequences. There's also what's called Qpcr. This is quantitative PCR, and it used to be called real-time PCR, where the nucleotides have little fluorescent tags attached to them, and with the inside of the thermal cycler will be a camera that actually detects fluorescence. And the more fluorescence occurs, the more copies of the DNA sequences have been made. And this is detected with the camera in real time, so that we look at, instead of looking at sequences, we will see a fluorescence signal start to rise with an increasing number of PCR product. And once that PCR product crosses a certain threshold, we now know that it's present. And we now know that it was present in the original sample. So that's done in real time, and we can actually watch the replication occur by fluorescence signal on a computer as it is occurring in the PCR thermal cycler. And there's a couple of more different types of PCR. I'm not going to get into all of them, but the general idea is that we have this general tool, PCR, where we replicate from a single strand of DNA into millions of copies of that DNA. And then we can take that tool and we can alter the way that it's used and use it in different ways. And I thought it would just be really it would be important and relevant to show you that this particular method, this PCR method, is what is used in our modern day genetic diagnostics. If you're interested in looking, well, what I may do here is I'd like to take you to this website, which also discusses PCR testing. Go to turn my pen off. We'll get this to open up. So this is another company that I looked at. I wanted to provide a link to you guys that is talking about the different types of tests that are used in COVID testing. So fighting the coronavirus here, these are all different types of tests that are used. We have antibody testing, which we'll cover in Unit 4. They can look for proteins and other antibodies. But here you can see we have PCR. We're going to look at what are called binding assays in the next lecture. ELISA, we will also look at an ELISA test in the next lecture, which is used for detecting proteins and antigens. And in these different testings, for proteins, we can use electrophoresis, but instead of DNA electrophoresis, it's protein electrophoresis, but the process is exactly the same. Here is PCR, and this is what real-time, so RT. Here we have high throughput, real-time PCR. And so they're looking for in vitro detection of the novel coronavirus. Here's another nucleic acid detection kit. So it's looking for reverse transcription, and this is referred to as amplification plot. This was that real time I was talking about in the fluorescent signals. So these are all different samples that are all being tested at the same time. They can test hundreds of samples at the same time if they needed to. So these are just different versions of different tests using biotechnology and immunology. By the way, we'll look at a microarray here that is used in diagnostic tools. And in this case, they're all being used in one very specific type of diagnostics, and that is in the coronavirus. So I hope that helped bring home some of the importance of these biotechnology tools. You will be in the healthcare industry, you will be using many of them and sending samples, and this is what they're actually going to be doing with these samples when they are testing them. So that's it for our PCR lecture, and in our next ones we'll talk about some blotting assays and some other genetic engineering tools. See you in the next one.